This is an OCR A level biology video on osmoregulation. We're going to be looking at osmoregulation, altering the permeability of the collecting duct as part of osmoregulation. So obviously looking at ADH, then we're going to look at kidney failure and renal dialysis, ways of treating kidney failure, and finally we'll finish it off by looking at urine analysis and pregnancy tests. This is following on from part 1 and part 2 on unit 5.2, and this will be the last video for this section. Let's start now. So osmoregulation is the control of water potential in the body and salt levels, making sure that at the right levels. So the correct water balance must be maintained between cells and surrounding fluids to prevent lysis, which is too much water in the cells, or crenation, which is too much water leaving the cells. So I'm going to draw a little table here, and it's water in comparing water out, so looking at the ways we can get water into the body and water out of the body. So some of the ways we can get water in the body is through food, so you just eat things, they contain water. Drink, that's pretty obvious. And respiration, so as we know, water is a byproduct of respiration, so metabolism also therefore generates water, and that kind of comes hand in hand with food. So ways we can get water out of the body is urine, sweat, exhaled air and feces, which does contain small amounts of water. The kidneys act as a balance to water levels in the body. So we've looked at all about the kidney, but we did brush over collecting duct fairly quickly. So ADH is a hormone which is produced to increase the amount of water reabsorption in the nephron, or in the collecting duct. So osmoreceptors in the hypothalamus detect low blood water potential and stimulate neurosecretory cells in the hypothalamus to release ADH. So neurosecretory cells are nerves which release these ADH molecules. So you usually wouldn't think nerves release ADH, which is a hormone, but neurosecretory cells do. They can secrete products. So ADH, how does this work? ADH is manufactured in the cell body and moves down the axon to be stored in vesicles, and these vesicles are found in the posterior pituitary gland. Action potentials from osmoreceptors cause these vesicles to fuse with the membrane of the posterior pituitary gland, and therefore releasing this ADH into the blood. So ADH will then travel through the bloodstream and meet the target cell. So, by the way, it's released by exocytosis, which I wrote there. Cells in the collecting duct have receptors for ADH. So ADH receptor complex, I made up that term, but that's what you'd usually say when a hormone binds with a receptor, you'd call it a complex. So an ADH receptor complex causes enzyme cascades or reactions. So vesicles containing aquaporins fuse with plasma membranes within our collecting duct, increasing the water permeability of the collecting duct. Therefore we get increased reabsorption as more water can move out, therefore decreased urine and more concentrated urine as more water has been reabsorbed in the collecting duct. Therefore less water goes towards the urine, so it's more concentrated. It still contains its urea, salts, etc. Okay. One thing about ADH, it's got a half-life of 20 minutes. So therefore, if we get a higher water potential in the blood, less ADH is produced. A half-life means basically it is broken down. So if we get low water potential in the blood, ADH is released to increase the amount of reabsorption of water in the nephron, in the collecting duct. This effect only lasts for 20 minutes basically, so the ADH then goes away, the aquaporins return into their vesicles, the collecting duct reduces its permeability to glucose, and its permeability to water, therefore less water is reabsorbed when there's high water potential in the blood, because we don't release ADH. So low blood water potential, we release ADH, increase reabsorption, and high blood water potential, then we do not release ADH, and the collecting duct does not reabsorb as much water. ADH therefore is controlled by negative feedback, as negative feedback is looking at reversing the change from the optimum. Okay, well, not from the optimum in this chance, the change in conditions, we just reverse it. So then we get if we've got a low water potential, we want to raise it again. Okay, so that is osmoregulation. Now let's look at different issues we can have with our kidney and how we can detect this. So first of all, I'm going to speak about the effects of GFR, which is glomerular filtration rate. So glomerular filtration rate, also said as GFR, urine analysis for protein and glucose can help indicate kidney failure. So what I'm going to draw here is a glomerulus, and it's a copy and pasted from the first slide in the first video, and during the copy and pasting we got some errors and these cells weren't copied, so I'm drawing them in here. So in the glomerulus we've got this afferent arteriole, which is wide, and the efferent arteriole, which is narrow. So a high GFR indicates a working kidney. A low GFR indicates a damaged kidney. Obviously there'll be an optimum GFR, and I'll try and find that out for you. So just a quick Google search, an optimal GFR is 90 or above, and kidney failure is a GFR of less than 15. Okay, so the kidney's just basically not working at that GFR. And something that can affect our GFR is the relative size of our afferent and efferent arterioles. So I think I watched a video on Khan Academy trying to explain this, and they did quite well, and they used the explanation of marathon runners. So if here we've got a huge bunch of marathon runners coming through this wide road, this is a really wide road, we can get loads of them through, call it a maze. Okay, so this is not how they did put it in their example, but I'm going to say we've got loads of competitors, a mass of people which are representing our blood, and they can then go into this maze. Okay, they can all easily flow through our different channels of the maze. Once they get to the end of this maze, they've got this really narrow channel, and they all take roughly the same time because the paths are the same sort of length. They get to this really narrow exit, which they have to go single file basically, and that's our efferent arteriole. So because this causes them to clog up here, the blood therefore stops here, accumulates in this area, and we get blood therefore resting on the endothelium of our capillaries. Therefore there's more time for diffusion between the blood endothelium, basement membrane, and epithelial tissue of the glomerulus in the podocytes. So more blood can diffuse out of this area. So efferent arteriole therefore slows down the movement of the blood here, increases the rate of diffusion. Afferent arteriole ensures lots get in. So what would happen if we made that the opposite way around? So that is just basically showing, the arrow showing lots of movement here, small movement here. If we did it the opposite way round, we'd get a very slow movement going into our maze. Okay, so single file, then once you get in, you can rush through the maze, and you've got a really wide exit because you're competing with no one. You've gone in pretty much single file. It took you ages to get in, but now you're in and you can just rush straight through. Now, exactly what would happen for blood. The blood gets in and it just rushes straight towards this wide area, passes over this very fast speeds, slow rates of diffusion, therefore as it has less contact with the basement membrane. Therefore we see lower rates of diffusion, so a lower glomerular filtration rate. Okay, and that is representing what I just spoke about there. Low filtration, low glomerular filtration rate. Here, wide afferent, narrow efferent, we have a high glomerular filtration rate, and that is how it is in humans. And one reason for a low GFR could have been these, but there's several reasons, but that's just one I'm going to mention here. I don't think you need to know it in too much detail. Just remember you have a wide afferent, narrow efferent. So let's have a look at some treatments for kidney failure, or just like not complete failure of kidneys, but damaged kidneys. They're not working as efficient as they were. And in general we have renal dialysis, which uses partially permeable membranes, a dialysis fluid which passes over the blood. It maintains high concentration gradients and the correct concentration gradient. So the dialysis fluid has the correct concentration of every single thing. If there's inadequate amounts in the blood, the nutrients diffuse in, so you might have inadequate mineral ions or sucrose or something, and they diffuse into the dialysis fluid. If there's excess, so it'll be urea, they then therefore diffuse out into the dialysis fluid, out of the blood. Okay, so it uses natural laws of diffusion. Waste diffuses out to the lower concentration in the dialysis fluid, because the dialysis fluid is like ideal things in excess really, and products the body needs diffuse in from the dialysis fluid into the blood. So there's two forms. We've got hemodialysis and peritoneal dialysis. Hemodialysis uses, first of all you need to use a drug called heparin, used to prevent blood clots. It uses countercurrent flow, so which you know in fish helps increase the rate of diffusion. Air bubbles are removed. It uses artificial capillaries, and you only need to use it 2 to 3 times a week, but you can be strapped to the machine for like 5 or 6 hours a day. Okay, we've been on a school trip and we've seen patients in the hospital using hemodialysis, and I'm not the best with that sort of blood and stuff, but I managed to sit through the hemodialysis bit, but not this peritoneal dialysis, which uses the abdominal membrane. So they pour dialysis fluid into an implanted tube in the abdomen, and there's natural diffusion between body cells and the cells and the dialysis fluid. They have to have a carefully monitored diet, and waste at every 2 to 3 hours is removed from this sac as it builds up. The sac is external, and you just literally bin it, and it's sort of like basically urine which is in this sac, or the waste that the body does not want. Finally, on this page, one other method is you can have a kidney transplant, and that's the most extreme method, but it can completely turn someone's life around. They could have a non-working kidney, they could be at risk of death, but this new kidney can just help them function like any normal person. They never have to get treatment after that, which is brilliant. But sometimes these kidneys can suffer an autoimmune rejection, so autoimmune response against the kidney, and it can be rejected by the body, so they have to take immunosuppressant drugs which significantly decrease the immune system and make the patient susceptible to various diseases that they would not usually be susceptible to. So it's not always the best option. Finally, let's look at some of the benefits of urine analysis. So we can analyse urine for glucose, alcohol, drugs, and molecules larger than 69,000 moles, which would indicate kidney failure. We can also use it for pregnancy tests, and let's have a look at how these pregnancy tests work. So HCG, which is a glycoprotein produced when pregnant. So a few weeks into pregnancy, this hormone HCG is produced. Monoclonal antibodies bind to this HCG. So first of all, urine is poured onto a test stick, and this HCG, if present, binds to the mobile antibodies which carry a blue bead. So these antibodies here. Mobile antibodies move down the test stick. If HCG is present, it binds to the fixed antibodies holding the bead in place, and a blue line forms. Okay, so if the hormone's present, these antibodies bind to this colouring region, forming a line. Okay, and if that line shows, it usually means you're pregnant. But there's also mobile antibodies used as a control to bind to the other fixed site to show that the test is working. So we get this mobile antibodies with the blue beads. They bind to the fixed antibodies and form a line if HCG is present. And just as a test to ensure that the pregnancy test is working, you have these unbound antibodies. They do not carry the blue bead, but they naturally just attach to the immobilized antibody. And if there is, and if they bind to it, and the blue beads bind to it, you get two lines. If you get two lines, that is evidence of a positive test. So anabolic steroids, how can we test for that? It's a huge thing in sport, a huge doping issue in sport, and the way they do it is through gas chromatography. So you don't have to know this in much detail. You just measure the retention time of the substance in this gas before it dissolves onto the chromatogram. So I found this on the internet. You've got this carrier gas which carries the sample, and a flow controller. So the gas is pumped at a steady flow through this column, and the sample is added. And the time it takes for the certain solutes to be carried in the gas and get to the end is recorded on this detector, which writes the chromatogram. So the gas dissolves into our chromatogram, and we get a result. And different heights of the curve indicate different substances, because each substance has a different retention time in this gas. Some take longer to diffuse round and they have a slower retention time. Others go round really quickly and they've really short retention times, and they indicate different substances, and obviously they'll compare this to other data. That is it on unit 5.2, happily. Really long unit. If you've not watched the other videos, just flicking through it, here's all the stuff I've done on 5.2. Thank you for watching.